And thank you for joining us today. We look forward to your presentation.
Okay. Hi, everyone. Okay, so as you said, my name is Carine Marcos. Thank you for inviting me. The aim of my talk today is not to give you a very detailed update on the field, but to give you an overview of the plant extracellular vesicle into context and what we have done, what we are learning from the past, and how we are looking into improving the field in the future. So a few disclaimers. There is going to be a lot of green in this slide, so if you don't like green color, I'm sorry about it. So this is a high-level talk addressed mostly at non-plant TV scientists, but if there are some people that are listening that are plant scientists, you will find it very low level because I'm not giving a lot of in-depth description, but it's to give an overview and provide context to why it is important to understand also what is happening in the plant EV field. I will discuss at the end the plant nomenclature. For the moment, I'm going to call these plant EVs or plant-derived vesicles, but I will give you a little bit of an explanation why it is important to name these vesicles with a specific nomenclature. So this is the structure of what I'm going to discuss with you. A little bit about comparing with mammalian EVs, the applications here, why it is important to understand the field of plant extracellular vesicles, a little bit of the timeline, some challenges we face in the plant EV research, and what the plant task force is doing and the aims of the task force in this field. So all of you are working on extracellular vesicles, so I don't need to give an update on what vesicles are and the clinical, biomedical, cosmetic, and other emerging applications of the mammalian extracellular vesicles. But you all know probably that plant extracellular vesicles are very similar to mammalian vesicles in terms of sizes, in terms of not the nature, but the fact that they hold RNA, DNA, lipids, and proteins, of course. But one of the big advantages of plant extracellular vesicles and plant-derived vesicles is that they also contain natural bioactive components, and this makes them very attractive for using these plant vesicles in different types of applications, clinical applications, biomedical application, cosmetic, and agricultural applications. And I'm going to go a little bit over each of them to give you an example. So you probably know that mammalian extracellular vesicles are currently being used in a lot of clinical trials, and these numbers are maybe not accurate because this is taken up by AI to see how many, but what you can see is that compared the mammalian extracellular vesicle clinical trials to plant extracellular vesicle clinical trials or plant-derived vesicles, there are really less plant-derived clinical trials than mammalian clinical trials. So we are about 10, 15 years behind the mammalian field, but they are starting to be increasingly popular because a lot of these plant vesicles have been shown to have some bioactivity on cellular extracts. And so many people want to now try on in clinical trials. And these are two examples. And of course, they use the term exosomes, but they are not necessarily exosomes, and we will discuss that a little bit later. But for example, plant-derived vesicles for patients diagnosed with polycystic ovary syndrome, or plant-derived vesicles loaded or not with curcumin to abrogate the symptoms of inflammatory bowel disease. So they are increasingly popular, and this is something that the plant field is looking into. The other application for plant vesicle research is in biomedical research. And this is taking advantage of the fact that some plants are already being farmed to express therapeutics for humans. And for example, Bentamiana type of plants are being farmed to express COVID candidate antibody, a vaccine for COVID. So this is increasingly interesting because of the low costs of producing these plants compared to mammalian cell culture costs. And so there are a few questions is, can we use these plant EVs for drug delivery systems? We use them cross-kingdom communication? And that has been shown for the clinical trials, for example. Can we optimize the EV production with bioactive compounds to express more of the bioactive compounds we want? One other thing is, can we engineer plants to generate EVs that are naturally loaded with therapeutically interesting drugs, RNA or proteins? Because you know that one of the issues in the EV field is, of course, the low efficiency of drug loading into EVs. And the plant field has the same issue, but we can use plants to engineer them and maybe naturally load EVs with these interesting drugs or RNA or proteins of interest. The other one is cosmetic applications, and these are some examples here. I didn't take them all, but they are available from the web. And you can see here, this is a clinic in London that is proposing plant-derived exosome therapy for facial rejuvenation, where they are giving you some plant-derived exosome cream. There is no where on the website where they explain how they are purifying these plant-derived exosomes and what they are exactly. They do say they don't contain DNA, but there is nowhere where they can explain how this is done. So there is a regulation that is lacking around this. And the field is going a lot into cosmetics in a lot of different countries, but the regulation is lacking. You can see here this product that is sold on the internet as well, that is sold on the internet with biostimulating cream and plant exosomes. And of course, when you try to see what are these plant exosomes or plant vesicles, you can't find anything. So there is a need to regulate all these applications. And of course, what we wonder is, why do we need plant extracellular vesicles in all these clinical or medical, biomedical or cosmetic application compared to mammalian extracellular vesicles? Because mammalian extracellular vesicles are well established. They are rich in cargo that is very relevant to humans. They have advanced clinical, we have advanced clinical experience with those, and they have the potential for personalized medicine. But what we know of that is that they have limited scalability. They are ethical and regulatory challenges, maybe safety concerns, because we don't understand exactly the whole content of these mammalian EVs, and there is potential immunogenicity from some of the EVs. And these are challenges that we don't necessarily find in plants if you use plant EVs, because you are exposed to plant EVs all the time when you eat food. And so they are known to have less immunogenicity. As I told you, they are easily scalable because of the plant farms. They are therefore cost effective, and they have a high safety profile compared to mammalian EVs. So they have a few advantages and of course a few disadvantages as well. But they might represent a few advantages if we can look into this a little bit more and try to understand if we can take advantages of these pros that they represent. So the other applications for plant EVs is, of course, the fact that they have a crucial role in host pathogen interaction, similarly to mammalian EVs. And there is a NIVI mediating a war between plant cells and pathogen cells. Here you can see where plants send plant EVs to lower the virulence of fungi or bacteria, and similarly, pathogens send fungal EV to lower the immune response of plant cells. So there is a raging war here, and we can harness this and adapt this and maybe boost the plant EV system to be able to engineer stronger plants that will resist pathogen invasion and will help improve plant growth and yield better plants, basically. So this is very important for plant health as well, not only for biomedical, clinical and cosmetic, but it's also important for plant health. As I said, response to infection, but also cell communication within the whole plant and cell wall remodeling, so adaptation to environmental factors. So we could also see that the application of plant extracellular vesicles is very important in terms of sustainable disease management, sustainable agriculture, maybe creation of alternative chemicals, eco-friendly EVs that are allowed to fight against pathogens without being dangerous chemicals, so better biological formulation, basically. So all these applications make plant extracellular vesicles and plant-derived vesicles very interesting compared to mammalian EV, so I hope I convinced you of that. And I thought it would be interesting to explain a little bit the timeline of discovery between plant-derived extracellular vesicles and mammalian EVs. So there was the first evidence of multivesicular bodies in plants already since 1965, with these multivesicular bodies fusing with the plasma membrane already in 1967. But between this time and now, it took more than 50 years to understand what these vesicles were. And it's only recently, in the year 2000s and further than 2000s, that plant extracellular vesicles are recognized as an entity, with a paper in 2014 showing that there is a mediation of plant-derived extracellular vesicles for interspecies communication. So this is the paper. I'm not showing you all the papers that have been published, of course, but I'm showing you critical papers that have been interesting for the field at that time. And that was one of the first papers that shows interspecies communication between plants and mouse gut host cells through edible plant-derived exosome-like particles. So you can see that they call that exosome-like nanoparticle, which is different from other papers. But basically what they did is that they extracted plant-derived nanovesicles from ginger root extracts. And I'm going to talk a little bit about the extraction protocols, but this was the first paper that demonstrated that these plant-derived vesicles are resistant to gastric and intestinal digestion, and that plant-derived vesicles could directly affect mammalian cell homeostasis. So that was a very exciting point in the field to show that there is cross-kingdom communication between plants and humans. And mice, so mammals. Then other papers here show different types of vesicles, and these are called apoplastic vesicles because they are purified from the apoplast, which is considered as the extracellular medium between the plasma membrane and the cell wall. And you can see that the first one to really establish plant extracellular vesicles from this was from sunflower seeds, and they were the first purification of these EVs in 2009. And then we had to wait 2017 and 2018 two papers from two different groups who have shown that extracellular vesicles carry stress response protein that are fighting against pathogens from the group of Roger Innes in Indiana, and the second group here led by Hailing Jean in Riverside have shown that plants extracellular vesicles carry RNA that can also fight against pathogen. So these are pivotal papers which have shaped the field into plant extracellular vesicle research. But you can see from the dates that this is much later than the mammalian field. The problem between these two and these few papers is that we are talking about different types of extracellular vesicles, and this has led myself with Laura and Marcella here, who have first isolated these sunflower EVs to write a paper in Jeff calling for rigor and standardization in plant extracellular vesicle research and asking for the distinction between extracellular vesicles, which are extracted from extracellular media, and plant-derived nanovesicles, which are extracted by other methods like soft tissue rupture, for example, homogenates, and might contain EVs, but might contain also other types of vesicles that are not EVs. And unless you purify them, they cannot be strictly called extracellular vesicles. So this is what we have published at that time to ask people to make the distinction. But you can see that although we are catching up, the field of plant extracellular vesicles is a little bit late, about 10, 15 years late compared to the mammalian field. And while the standardization in MySafe came in 2014, we are just about now in the plant EV field to form groups to allow to ask for standardization across different groups. And so what are the challenges that the plant EV research faces specifically? And I think most of you, if you are not working in plants, the first thing I am told is that the first challenge is the cell wall. And indeed, that's one of the challenges, of course. And you can see here this paper in 2010, I think, where you have the cell wall here and extracellular vesicles being released in the cell wall. And what we are learning and starting to understand is that the CVs actually contain enzymes that can modulate the cell wall. So the cell wall is actually a small issue, but not a complete issue also because it depends on the type of plant you are using to extract your extracellular vesicles. Also, we have the field has developed a specific technique that is known as infiltration, where you actually can extract EVs without degrading the cells. And this is using vacuum, where you impose a vacuum either with a syringe or a vacuum pump to expel the air from the extracellular media. And then when you reimpose the vacuum, you can have the liquid going and filling the extracellular space in these leaves. And this allows the EVs to then be collected by centrifugations, so of the leaves that have been infiltrated. So you can see here that's a leaf that hasn't been subjected to infiltration yet. And then you have a leaf here that has been subjected to infiltration. And you see these dark green patches. They are the regions where the leaves have taken up the infiltration buffer that is then going to be collected and release these extracellular vesicles in them. And this fluid is then called, in this case, apoplastic wash fluids. And this fluid is what you are going to use to centrifuge and to collect your EVs. But this is, of course, only when you are studying leaves. And this has been done very efficiently for Arabidopsis thaliana, which is a typical plant model, where the leaves are very small here. So you need a lot of these plants to purify enough EVs. But if you are studying another type of plant like ginger roots, olives, lemons, tomatoes, or sage. Sage has leaves, as you can see, and you can touch them. They are completely different from any type of plants. For example, spinach, we have very thin leaves, and so these sage leaves might not be available to be infiltrated with the method I have sent you. So actually, the main issue for the plant researchers is the variety of plant sources that is causing a problem, because we can't compare studies of EVs done with tomato juice, for example, and done with olives or gingers. So that is a very big issue, the diversity of the biological systems in the plant field. So this leads to a variety of techniques, while in the mammalian field you have, of course, the cell cultures, but you have also fluids like blood, milk, etc. In leaves, you have so many different types of sources like juice, homogenates, roots, cell culture from roots, leaves, plant cell culture, or the extracellular media that is called the apoplats, as I told you, which will lead to different types of purification. So when you then centrifuge these liquids that you get from all these different techniques, you will have different mixes of extracellular vesicles. So not only different plant sources will give different types of EVs, but they will give different composition and different chemicals in there, different bioactive molecules. And on top of that, in the fields, we also have historically EVs that have been separated at 40K instead of 100K. Although the mammalian field currently uses 100G all the time, sorry, not kg. The plant field is currently debating on whether the 40,000 G and the 100,000 G pellets are similar or represent different types of extracellular vesicles. And we seem to agree now on the fact that they represent different types of extracellular vesicles. And we are currently trying to understand a little bit more and to characterize these types of vesicles. So the fact that we have so many different types of plants and different techniques makes it very difficult to standardize the field. And this is one of the example that I can give you, where we published in 2024 a protocol for the isolation of small extracellular vesicles from the apoplastic wash fluid of Nicotiana bantamana leaves. So all the protocols that have been published before that were on Arabidopsis, which I've shown you before. And this is a plant that is completely different from Bantamana. It's very small with very small leaves. Bantamana is this type of plant. It has very big leaves, and it's currently a plant that is used in pharmaceutical settings to express proteins for example, for vaccine, as I told you. So we had to we have noticed that using the same protocol as Arabidopsis didn't work. So we had to come up with a different protocol, adjusting the centrifugation times, adjusting the infiltration buffers, adjusting different rotors, making sure when you infiltrate, you don't break the cells, which ends up by having a fluid that is green. That means that the cells have been broken and you have actually chloroplasts in there. So making sure that you have a clear apoplastic fluid. So all these had to be optimized, although we already had one that was working for another type of plant. So you can imagine that every scientist in the world working on a different plant will have to adjust their protocol to their type of plants. and the type of EVs they want to purify. And this makes it really difficult for the plant field to homogenize and to compare the different studies between different scientists. But we have managed to agree on some things, and the fact that there are different extracellular vesicles in plants. Here, this publication gives you 4 examples of this. These Expo positive EVs and ATG8 positive EVs are specialized EVs that are being studied, and we don't know a lot about those. But these 2 are mostly interesting for the mammalian field because these vesicles here are supposed to be labeled by proteins that we call TET8 and TET9. These are tetraspanines, similar to the mammalian CD63, for example. And they are, and these markers are colocalizing with markers of the multivesicular bodies, which suggests that they might be of an exosomal origin. The other type of plant extracellular vesicles that come up at a different density is marked by the protein called PEN1, which is also called CYP121, which is a syntaxin. And because this marker doesn't colocalize with markers of the multivesicular bodies, we think that it can be a different type of EVs, not from exosomal origin. So the field is currently looking at all these type of EVs and trying to understand what is the composition in lipids, in proteins, and seeing if these markers that have been identified in Arabidopsis thaliana are also markers of other types of plants, which can be the case in sometimes, for example, in Bantamyana, but can also not be the case in some other type of plants. And so you understand that at this point, we have to find yet again new markers and again prove the origin and the purity of these EVs in different types of plants. So all these, of course, impacts on the type of vesicles, the composition of the EVs, the purification methods, and the protein markers, because even if you have protein markers that work in one plant, for example, in Thaliana, they might not cross-react with another, with the same protein in another plant. So that makes the standardization very difficult. In the mammalian field, when you have a CD63 antibody, you know that in one cell type, it might work in another cell type. But that's not necessarily the case for plants. So this is delaying the field very much because we need to develop much more tools than the mammalian field is currently requiring. We need to develop each tool for each of the plants we are using. So this is why we have started the plant task force, and I'm going to explain to you a little bit about this task force, the aim of the task force, who is working in this task force, and what we are trying to achieve. So Gabriela here, he's a researcher in Italy, and together with Annie, and I couldn't find a picture of Annie, sorry, they have set up the first symposium on plant extracellular vesicles in Vienna in ISF 2025, where they invited some experts. Among them, there was Alfredo here, Fernando, Ronel, Baue, and Antonio to discuss and exchange ideas around groundbreaking research around plant EVs, discover the EV function, and trying to connect all the leading plant EV scientists across the world. So that went really, really well. And we decided to set up the plant EV task force here. And you can see here, that's a photo taken at the time. So you see, we are not many at the moment. And this is our LinkedIn page. And in the task force, we want to try and get people together to understand how we can help standardize and communicate between groups and make sure we can compare bioactive EV research from one group with another. So at the moment, we are around 20 active members, and we meet every 2 months as a task force. but we also have 4 working groups that meet ad hoc and report to the task force. We have recently set up a LinkedIn page, a next page, Instagram and Blue Sky, where you can see some updates. And if you want to, and if you are interested in plant TV research, I invite you to follow us on the platform of your choice. So what are we doing? So as I said, we have 4 working groups. We have working group 1, which is the nomenclature, which is led by me. I'm also the co-chair for the task force. We have a working group 2, which is focused on education, dissemination, divulgation, which is led by Fernando de la Cuesta in Madrid and Stefania Raimondo in Palermo. We also have a working group 3 on bioactivity, which is led by Alfredo Ambrosone in Italy and Ron Elroth in the UK. And a working group 4 on exploitation, which is led by Stefania Raimondo in Italy and Alberto Davalos in Spain. So what are they doing? The working group 2 here is focused on education, dissemination, and divulgation. And they are working on public engagement, setting up material for conference and satellites, on dissemination, on preparation of portfolio and media engagement. And at the moment, we have recently posted a video from Gabriela that you can see on LinkedIn and on all these pages, where we can see we're going to launch a few videos to explain what we are doing and how important the task force will be in homogenizing the research in plant extracellular vesicles and plant-derived vesicle research. The working group 3, led by Alfredo on bioactivity, is tasked with standardizing purification procedures across different types of plant-derived vesicles and plant EVs. They are tasked by trying to find a regulation on bioactivity definition. So, for example, if you have vesicles from pomegranate or vesicles from ginger, how do you compare the activity? Is it just in number of vesicles, or can you compare it with something different? They're also tasked into looking at homogenizing biomedical applications and agricultural applications. The working group 4, exploitation, focuses on evaluating and advancing the practical scalable applications of plant EVs by facilitating the transfer of knowledge between academia and industry, promoting best practices within the task force. So you will understand that this working group is working closely with the other working groups, and they are identifying companies and maybe analyzing the regulatory landscape for novel food and cosmetics, and engaging with stakeholders and industry, and working closely with the working group 2 for dissemination. And this is early stages. That's the plan. Nothing has been set up yet into motion, but this is the plan we are doing. I am currently leading the working group 1, which is on nomenclature, because if you look at the plant extracellular vesicle and plant-derived vesicle literature, you will find many different terms. Among those, you will find plant extracellular vesicles, PEVs, plant-derived exosome-like vesicles, PDALVs, plant-derived vesicle-like nanoparticles, plant nanoparticles, plant-derived extracellular vesicles, or exosome-like nanovesicles. So all this is confusing to the field. There is a lack of understanding how we can compare all these, and of course, together with the guidelines from MySev, which are not recommending the use of exosome unless you have demonstrated an exosomal biogenesis pathway. We need to homogenize and standardize all this. So we are working on that at the moment. We have about 10 members in the working group who are focusing on this. We are going to launch this month a survey on nomenclature that we are going to open to all scientists across the world who are working on plant extracellular vesicles and plant-derived vesicles. And the aim is to write a white paper and to propose a nomenclature that will be standardized across the field so we can all understand how we can move from just naming DCVs at the moment and then moving on to standardizing purification procedures, bioactivity, etc. So the overall aim of the plant task force is to accelerate the research on the plant EVs, establish standards and best practices, enabling translational applications, and promote global collaboration and innovation, very, very much like MySafe did, but with the specific challenges that the plant researchers face. And this is it for me. I hope I gave you an overall view of the field, and I'm happy to take your questions and have nice discussions about what you are looking into and what are your concerns and challenges and what you are interested in this field.
Yeah, thank you so much for this comprehensive presentation. Yeah, it's a very promising field, yet it's so challenging. From your presentation, I can sense that. So we have one question at the chat box from Yudai Yamakawa. In the context of a plant EV supplement, what are the benefits of isolating the EV from a plant and intaking it versus just eating the plant directly?
Yes, it's the concentration that is the difference. When you are eating it, it goes through your gut, and then although for ginger it has been shown to go through, some scientists are questioning if this is correct or not. So one of the benefits is that you can maybe take this in different forms, but also it's a matter of concentration. So if you take it, of course, every day and you eat it and it has a beneficial for your health, it's amazing. And we know that this is true. But sometimes if you concentrate and in specific pathological case, you might help increasing these benefits.
Yeah.
And does that answer your question?
Yes, thank you. Yeah, any more questions for Dr. Karen? Okay, maybe I'll ask a question. So what's your, I can see the field is so challenging, especially that there is, how to say, as you mentioned, like comparing between the mammalian EVs and the plant EVs, it's so difficult to standardize. For example, for characterization, there's no one protein, for example, marker that we can rely on to differentiate the EV from non-EV particles in terms of the plants. So how do you see the field moving forward? I mean, addressing this challenge?
Yes, it needs a lot of collaboration, and it's difficult because everyone is set in their ways, and it's normal because everyone has their babies. When you start working on something and you start to name it and you start to look at bioactivity and the effect on health is you don't want to change, but it's important to collaborate, and that's the aim of the task force, to see how we can make sure we homogenize this by talking through the pros and the cons, because you will know the advantages of your specific system. And if you work on a specific system, I might not understand the advantages of what you're doing. So it's important to discuss, understand each other's pros and cons of using this technique, using this plant, using this concentration, using these particles, and then seeing what we can do to homogenize and compare. But this is going to be difficult, and that's why we are starting by the nomenclature first. And then we will go into maybe purification types. And this has been ongoing discussion as well with the mammalian field, as you know, it has been going on for 10 years. So I see that this is maybe going on for a few years as well for the plant field. But we are lucky that we have a community that is very open to discussion. And so I think we are going to go a little bit faster than what we were expecting, maybe with the mammalian field. At least that's what I hope.
Yeah, interesting. Yeah. So Yudai Yamakawa is asking, does the use of pesticide affect the output or the profile of the EV, of the plant EV? The use of pesticides.
Well, that's a very interesting question. I assume it would. I don't think we have evidence yet to show that, or at least I don't know of any evidence. But I assume it would, because when you use pesticides, you change some physiology of the plant as well, hopefully not too much, but it might be reflected in AEVs. So this is something definitely that we need to look into.
Okay, thank you. So Heather is asking, is there any way that students can be involved in supporting task force initiatives if they are also interested in plant EVs?
Yes, very good, Heather. We are open to anyone that is interested in supporting the task force and being active in the task force. So we maintain a close group because if we start by 100 people, it's difficult to discuss with 100 people. So we maintain at the moment a small group, but there is some people that are leaving because they don't have enough time, and so we are taking regularly new people that are very active. So if you want to be involved in a task force, you can write to Gabriela. You will find her email address on internet. You can send her or me your interest, and then we'll send you a form, and we will see when we are taking inputs. Then you can certainly join. So please send an email with your interest, and we will make sure you can participate if you are active.
Yeah, great. So here a question, are plant-based EVs more effective than eukaryotic-derived EVs?
That is an interesting question, and that is something that we are looking into, but it depends what you call more effective. If you are looking at, for example, let's say you are using plant-derived EVs and mammalian-derived EVs effect in a specific cellular function in a cell culture context. If you are using plant-derived EVs, you might have bioactive molecules that you don't have in mammalian-derived EVs, so you might have different effects. So I guess it depends what is the effect you are looking for. Is very difficult to compare. So if you're saying in terms of if your question is about drug delivery, so if you have curcumin, for example, or any other drug, and you're looking at whether a plant-based EV is more efficient at delivering that particular molecule into human cells than eukaryotic derived EV, then this is something you need to assess because it will also depend how much you load of this drug on UVs, what is the efficiency of loading then. And eukaryotic EVs might be more efficient at delivering because they have, of course, molecules and proteins that are more compatible with mammalian cells. So the answer to your question will depend on what exactly you are looking into.
Yeah, and I saw some studies like they are merging the effects of the mammalian EVs and also the plant EVs. So they synthesize these hybridized EVs to gain the benefit of both sides. Yeah.
Yes, there is that hybrid IVs, and there is also a lot of hybrid IVs in terms of using plant-derived EVs and mixing them with lipids. So there is a lot of potential that can be achieved with plant EVs and plant-derived vesicles. But we are just at the infancy, and we need to have a lot more research on that and define what are the outcomes you want to see, which relates to the last questions.
Yeah. So, Dr. Nur Hayati, you want to unmute?
Hi, hello, Carine. Thank
Hello.
you. I'm Nur Hayati from Malaysia. You for sharing this topic today, because I think we in Malaysia we are slowly moving towards plant EV research. Beside me, Dr. Uy Dijun also doing plant EV research. So the great effort from the ISAF task force for the plant EV task force team, putting everything in group so that we can understand and we have reference for our research to go on. Hopefully in the future maybe we can have a confirm a guidebook, maybe some kind of guidebook or anything in this plant EV research, so that we can move forward to expose the benefit of potential benefit of plant EV in research. Yeah, thank you for today's sharing.
Yes, I agree with you. I think it's important. And I think it's important as well. And that's why I'm leading the nomenclature one, to make the difference between the plant-derived vesicles, which are the vesicles that you get from all these medicinal plants, for example, that are homogenized or from where you extract the juice, and the plant extracellular vesicles that you obtain specifically from extracellular media, so like conditioned root media or apoplastic media. So this, if scientists, if we can agree on a way forward to name this, then we can already have a step to bounce on to then study even more the effect of all these types of vesicles on human health.
Dr. can I add on a bit more?
Yeah,
Do you
of
think
course.
that, okay, now like we are using the plants, the leaves or the fruits, do you think that when we do plant cell culture, is it more beneficial so that we can get consistent EVs, or have you ever tried?
I have not done EVs from cell culture, but I know a few people who have done it, and it seems to be more consistent, of course, because it's the same as mammalian cells. You can standardize and make sure your conditions are exactly the same between different preps, so that might help. However, because it's not a biological system, natural system as such, the cell cultures from plants, it's an artificial system, right? So that means that you might not have so much benefits compared to other types of systems. But I think all systems are very interesting to study at the moment, because we are at the beginning. And we can probably, if we understand both systems in parallel, we can probably engineer this plant cell culture to then produce a lot of very homogeneous types of EVs that might be producing therapeutic compounds that we are interested in. But at the moment, we don't know yet, right? So it's a balance between same conditions, same purifications, and the bioactivity of what we want to, of the bioactivity of the EVs that you are interested in.
Yes,
Thank you, Rada. Thank you very much.
Yeah. I hope you agree, actually.
Yeah. Okay, thank you.
Yeah, so and before we end, I would like to ask about what would you advise a new researcher, an early career researcher who is interested in this field and want to start doing the experiments and try plant EVs? What would be your advice?
I would say a good thing is to first read the publications that have been there, but also contact the researchers themselves because I'm sure they are always happy to discuss techniques and purifications and pros and cons and what has worked for them and what hasn't worked for them. Because some of the things, for example, we have worked in my lab, we haven't published because it's a negative result. And then someone starting might start on that thing. And then we can say, well, actually, we tried that, it doesn't work very well. You might be able to tweak it like this, and you will improve your yield, or do a little longer centrifugation or things like that. So I'd say contact people, talk to people, mostly in ISF meetings or in this type of talks. Make sure you discuss openly, because that will help. We are the advantage with the plant field is I don't think we are in because we all have a lot of different systems. We are very much collaborative in this field. So I think it helps a lot when you are new student who wants to start, because a lot of the researchers are very accessible to questions.
Yes, yeah. So thank you so much for today's talk and for all the information. It was so enlightening and fascinating. And wish you all the best with the task forces and your efforts to bring everybody together and try to to help this field. Yeah. So thank you everyone for attending and for joining us today. And yeah, have a nice day or good night in some places as well. Yeah, thank you so much.
Yeah, and thank you very much for inviting me. I think it's an excellent initiative that you are trying to see different types of EVs. And feel free to send me an email if you have another question. I'm happy to answer anything. And thank you very much. And good
Thank
night
you
or
so
good
much.
day.
Thank you.
Bye.
Bye.
Thank you. Thank you, Dr. Carine. Thank